So how do the batch correction methods work? Well, let's start with the most harsh one, the Seurat Canonical correlation analysis-based correction. So it will take all the cells of, in this example, the two samples and calculate correlations between the cells. And the correlations are calculated based on the expression levels of all the genes in each cell. So hopefully it will find cells that are very correlated, and it's going to use these cells as anchors for doing the batch correction. So cells with high correlations are considered anchors, and he favors the anchors that have very similar cell neighborhoods. You see that he finds anchors here, so cells with high correlations. And then he will look at the vicinity of these cells to see if he can find similar cells in the vicinity. And if this is the case, he's going to favor these anchors. And then he's going to use the anchors to calculate correlation factors. So as you can see here, these cells and these cells, they are in the UMAP on more or less the same place. They have the same cell neighborhood. So he's not going to do a big correction here. But for instance, this cluster will find anchors all over the UMAP of the other sample. So here he knows that he has to do a big batch correction. So the correction that he does can be very harsh. So it can change the data very much. That's what we mean with harsh. So you typically use this when you have very strong batch effects. It assumes that the clusters that are found in both samples are very similar, so that you have the same cell types in the two samples that you are correcting. So you typically use this with strong batch effects, and examples of strong batch effects, and I will go back to this slide for this, are when, for instance, you try to combine data from different species, or you try to combine single nuclei and single cell data, or you try to compare different tissues. But here, this case, it has a strong batch effect, but this will not work with the Seurat CCA method, because when you compare different tissues, you expect to have different cell types, and then CCA doesn't work. Strong batch effects are also when you try to combine different platforms, so for instance 10x Genomics and SmartSeq data, as is the case here in this example that I show here on this slide. So these are all different platforms, and you see that they produce a very strong batch effect.